A lot of support. I also want to say this is the greatest conference on the planet. I don't like calling it a conference because it's a festival. I speak at like 20 plus conferences every year, and this is the one I look forward to the most because it's got the best crowds, the best speakers. So what I'm going to be giving an update today is telomere science, where we are right now. And I don't have a lot of time, so I'm going to start off quickly with something I always say, and that's bad things happen when telomeres get short. And that's, if there's anything else you don't learn in this, this is what I want you to remember. Bad things happen when telomeres get short. There's nothing good about short telomeres, and there's everything great about long telomeres. Okay, so we're getting to this, so what is this telomere, this word I just said? A lot of you have seen this before, but some of you haven't, so I'm going to go quickly over the basics of what telomere biology really is. And to do this, we need to zoom in on a human being. And when we zoom in a lot, we see that a human's made up of 100 trillion cells. I mean, telomeres, there's a lot of telomeres in us, and they're very small little things, so we got to zoom in really deep. But most of the theories on why we age say that we age because our cells age. So this is, in my research, this is where we target. We look at the cells. We got to zoom in further. We see that every nuclei contains chromosomes. These things shown in blue. Chromosomes are where your genes are. They give you your hair color, your eye color. And we got to zoom in on one of these chromosomes now. And we see that a chromosome is made up of two arms, a top arm extending all the way from left to right, and a bottom arm extending all the way to left and right. Inside of each of those arms is our DNA. DNA is a long string of beads. A typical DNA molecule is about 100 million beads in length. The beads are called bases, and the sequence tells us what our hair color and eye color are. But I want you to think of these as shoelaces. Long, long shoelace. And it's so long that it's all coiled up like a slinky inside these chromosome arms. And everybody knows that our shoelaces have these caps on them. And these caps protect our shoelaces. Well, that's what telomeres are, shown here on the end of these chromosomes. Telomeres are there to protect our chromosomes from degradation and all kinds of different adverse things that can happen to them. Well, let's now zoom in deeper, and we'll zoom in on one of the telomeres. We'll unravel that slinky that I had mentioned. And we find that a telomere is about 15,000 bases in length. Remember I said the human chromosome average is about 100 million bases in length. So telomeres are pretty small things on the end of our chromosomes. They're 15,000 bases at least when we're first conceived. And here's where all the troubles begin. This is why I'm doing all this research and stuff, is because every time our cells divide, each and every time our cells divide, our telomeres get a little bit shorter. And there's so much cell division to go from a single cell embryo to a newborn baby that by the time we're even born, our telomeres have already shortened down to 10,000 bases. This is not a theory, this is recognized fact. Anybody who does any work with human cells growing in petri dishes and labs all over the world know this. This is not even in question anymore. Our telomeres shorten with cell division. Well, it doesn't stop there. It still gets worse because we got a lot of growing up to do, a lot of cell division. With more and more cell division, the telomeres get shorter and shorter. And when our telomeres get down to 5,000 bases, we die of old age. Our cells lose the ability to function, and we can no longer live. Our skin dies, our immune system dies, lots of different cells die. Let me just review that again, because this is really important, is that people want to understand telomeres. We are conceived at 15,000 bases. We are born at 10,000 bases. And we die of old age at 5,000 bases. And there's absolutely nothing we can do about this yet. We can slow it down a bit by healthy lifestyles and stuff like that, but we can't stop this or reverse it. But I'm going to make it so we can. That's what I'm doing. And it's the cause of almost every disease you've ever heard of. Almost every disease you've ever heard of has now been published in scientific peer-reviewed journals showing that the length of the telomere plays a role in doing this. Okay, I want to lengthen the telomeres. That's what my whole research is about. I want to lengthen them, and the way to lengthen them is using an enzyme called telomerase. Telomerase this is showing a cartoon of telomerase. The green squiggly thing is the DNA molecule. The enzyme itself is shown as a factory up there. And you can see it's adding little green things to the very tip of the chromosome. Those are new bases or new beads being added to lengthen the telomeres. So that's what telomerase does. Telomerase lengthens telomeres. Okay, so that's great. Now how are we going to get all of the cells of our body to contain telomerase? Telomerase has to get inside of our cells somehow. So how are we going to do that? And that's through a process called gene delivery, also called gene therapy. What I'm going to do is I'm going to explain what this process is all about, because it's being used a lot right now. And to do this, I want you to first think of a human cell as a soap bubble. So we have a human cell that's a soap bubble. And believe me, those cell biologists know that really is. We are a lot like soap bubbles, our cells are. Okay, now I want you to imagine another soap bubble is blowing out this thing, and it's sitting right there. It's a little smaller. And now imagine that we now put the gene for telomerase inside that soap bubble. The gene for telomerase is what produces telomerase. And this particular gene that we put into the small soap bubble is on a super on switch. I mean, it produces telomerase at higher levels than you would ever see anywhere. Well, we all know that when you're blowing soap bubbles and they're floating in the air, and two of them fuse, it goes. When two of them fuse, the contents of each become one. That soap bubble becomes one little slightly bigger soap bubble, and the gene gets inside and produces telomerase, now shown here just as a factory without the DNA. And this is on such a strong on switch that it ends up producing lots of telomerase. Okay, and this is what we want because we need lots of telomerase to actually lengthen telomeres. This technology is not new, just new things have been developed lately that make it a little safer than the old methods that used to be. But so far, we have reversed aging in human cells in vitro. We have reversed aging in human skin that was grown on the back of a mouse. So we've done a tissue also. And scientists at Harvard have now reversed aging in engineered mice. I say engineered mice because mice don't normally have telomere shortening. They're different than humans in that way. But people have made mice that do age the same way as humans do with telomere shortening. And they have reversed aging in these mice. They turn old mice into young mice. And what's really exciting is it wasn't just appearances, it was everything. Their ability to breathe, their ability to remember how to go through a maze, memory came back, things like it made people start asking, wow, is Alzheimer's and other forms of dementia a loss of memory? Or maybe it's just a loss of access to the memory. And maybe lengthening the telomeres has restored that access. So when I say reverse aging, that's with every biomarker of aging that was looked at, not just increased energy levels. They saw a reversal of aging in almost every way imaginable. So let's do humans. We're ready. Let's start doing humans. Well, we already have, and you all know her as Liz Parrish, the very first person to ever receive this treatment. I would say that she's a lot braver person than I am. I'm impressed with her guts. I would say that 95% of the world that knows about gene therapy knows about it because of Liz Parrish. And she'll be up talking in a few minutes after a few speakers after me, so I really look forward to hearing her, as I hope you do too. So, last year at RAADfest 2017, I met a man named Jeff Mathis. And Jeff, where are you? I'd like you to stand up. There he is, way in the back. That's Jeff Mathis. He came to me and my partners and he said, I want to make this happen. I want to see gene therapy actually get through clinical studies and get to be practiced. And he didn't understand why nobody else had done it. He's got experience. I should have said Dr. Jeff Mathis. He's got experience doing this kind of stuff, and he said that he could have this done in a short time. He said, wow. So we all agreed. We said we'd do it. Well, why hasn't Labella treated anyone yet? It's been a year. But what's the bottleneck? I'm the bottleneck, okay? Jeff and the rest of the team at Labella have been doing amazing results, getting a lot of things set up, which I'm going to explain in a little bit. But there is this one thing, is that if somebody that dies in a clinical study or gets really sick, everybody's going to look at me. They're going to point their fingers at me. I'm the guy, okay? And so it kind of puts me in a kind of different position than most of you would think you would be if you were in my position. I got to make certain I'd be devastated if somebody got really sick or died in the study. My goal is to help people, not hurt people. So I'd already read some clinical studies and stuff like that from other people doing the gene therapy. And I was a little concerned about things, but I thought I'd spend a few months just reading everything I could, all the clinical studies, going to conferences, meeting the world's leading experts in gene therapy, and find out what's really going on. Because this thing on the right, the small bubble, that's called the vehicle. And in the clinical studies, that's the problem. Not the cell, not what's inside the vehicle, but the vehicle itself. In 113 clinical studies, it had no serious side effects. That's predecessors to this type of bubble did have a lot of problems, but this one was the first one. It didn't have any serious side effects, and that's what all the publications were saying. And it was great. They were talking about all the great things about it. But it was kind of misleading because they were also saying in small print, well, except some of the patients had immune sensitivity. So when I researched further into this, I found that the immune sensitivity that's being seen is something called a cytotoxic T-cell response. The cytotoxic T-cell response is something really bad. This is where your immune system starts attacking your cells and killing your cells. And in our case, it will kill every treated cell. And if we want to do a gene therapy to deliver our gene to every cell of the body, and then that person develops a cytotoxic T-cell response, that person is dead. Okay, this posed a real problem to me. But as I said, I spent months reading all the clinical studies, went to conferences, talking to the world's experts, and as a result, I've made some good connections with some people that have some very promising data that very few people know about, and we have now solved this problem. We will not have anybody with a cytotoxic T-cell response. Great. Now that wasn't the only problem. So we had that problem solved, but we also saw in the studies that 70% of all the people that volunteered to be in the study were excluded. They were rejected from being in the study because they contained some type of substance. Sometimes it was antibodies, sometimes it was something else. Neutralizing substances, they were called, inside their blood, which meant that as soon as they put the gene therapy into the blood, their body destroyed the gene therapy before it ever got into any cells. So it was a complete waste of time. So a lot of people can't be treated. But we solved that problem too. And I mean, there's some great science going on. We didn't solve it ourselves. It's been collaborating with other labs where this has been solved. Another problem is that we learned that the DNA sequences that were put into cells actually have components in them that actually induce an immune response. And so we had to go through and remove all these sequences, change it into something else, still have a very active gene therapy. And we solved that one too. This is all in the last year. I want to, even though we haven't treated anybody, I feel like we have made major progress. And I think we're really on the forefront here. One other problem was, it was found that whenever anybody got treated in a clinical study, and all these clinical studies, they were not delivering telomeres, they were delivering other genes. But whenever they got treated, it turned out they could not be treated again because their immune system got so powerful against that gene therapy that they could not be treated again. So people could only be treated once. We solved that problem too.
Wow, wow.
And so I said to Labella, go for it.
All right.
I'm not standing in the way anyway. They were excited because they were getting pretty upset with me for being so worried about, hurting somebody. But they didn't want to hurt anybody either, but they didn't realize that there was so much involved yet. But I still want to ask the question because if somebody died or somebody got sick, I would feel like, well, I did my best, okay? And that says something, but I'll still be devastated if somebody dies. But I still want to ask the question, is telomerase gene therapy safe, and when is it okay to do it? And the big question that I ask myself is, when is the risk of no treatment greater than the risk of treatment?
Exactly.
And that is really important. And there's two diseases that I want to mention right now in particular that are always that way. And one is Alzheimer's disease, and the other is extreme old age. Now, we probably all know from experience, knowing others, that when you get diagnosed with Alzheimer's, you typically have about 6 years left. Okay, so it's a death sentence. Nobody survives. It's a 100% fatal disease. Extreme old age. Do you know that if you are a very healthy 89-year-old, statistically, you have an 11% chance of dying in the next year? I mean, that's pretty risky, just being extreme old age. And so these are kind of people that I want to see get treated right away, especially now that we think it's safe. So the most serious risk to the patient, in my opinion, is not treating the patient, especially when they're suffering from one of these diseases.
Yeah.
Thank you. This is a list of all the telomere shortening diseases that were published probably like 10 years ago. Now this list has gotten so long that I don't know of a single disease that's not connected to telomeres. But when you look at this list, the most dangerous thing on there besides aging is Alzheimer's. And so that's a disease that we should try to work on. Labella Gene Therapeutics is now doing an Alzheimer's disease clinical study. They have now set up labs, or are setting up labs in Colombia, Vanuatu, spelled that wrong, sorry, Vanuatu, and Singapore. They're very close to having everything ready. We have already identified our first subjects. Subject 2, as you know, Liz Parrish is subject 1. Subject 2 is a 72-year-old male that is suffering from advanced Alzheimer's. He was diagnosed 5.5 years ago, and I already said that most people die after about 6 years, so he's close to really needing our help. We're scheduled to be treated on December 10th, 2018. Subject 3 is suffering from early Alzheimer's. He was diagnosed 2 years ago, and he's going to be treated in February 18th, 2018, or 19, sorry. They're giving me a timer here, I gotta go faster. Delivery will be intravenous and interthecal. There will be numerous aging and cognitive markers will be measured throughout the study, baseline and at 1 year. Telomerase activity and telomere length measurements will be done at baseline, day 1, day 3, day 9, etc., up to 1 year. And we will be following these patients for the entire rest of their lives. The major questions we're going to be asking is, did telomerase gene get delivered? That's a big question, because if they have something neutralizing substances and it got destroyed, it doesn't count. Even if we think we have it solved. Did the cells produce telomerase? If the cells that got it delivered, did they actually produce telomerase? And then if the cells that produced telomerase did get telomerase produced, did the telomeres get longer? Well, we've got to ask all those 3 questions before we can ask, did this provide any benefits to the human? It doesn't matter if we give them telomerase and it doesn't lengthen telomeres. So our most optimistic speculation is we'll have the results by February 2019. That's the most optimistic. But keep in mind, a clinical study is a basic research study. And we are going to be learning because we're going to be asking these 3 questions first. And if the answer is no to any of these, we will have to figure out a way to make that part work. And we already know what to do. So it's not going to be too long. But we got to do that before we can answer that last question. Did this provide any benefit? humans. I didn't have time because it says 12 seconds left here for me now, but one of the subjects I do like to talk about is telomerase and cancer. There's a rumor going around that telomerase causes cancer. Instead of proving it to you today, this book of mine describes it in great detail. Will prove to you that that's just a false rumor that's made up by people that don't have that strong a background in cancer. It's available in English, Chinese, Japanese, Korean, and Spanish, and it's available at defytime.com. One other announcement is that a coin has been launched by Defy Time called the Defy Time coin. It's available at www.defytime.group, but it'll be listed in early October, so that's in less than 2 weeks, and it will be used to fund telomere research. And I want to end with something kind of interesting. People always ask me, why do I want to live so long? Am I afraid of dying? And my answer is, no, I'm not afraid of dying. I'm afraid of missing out. There's a lot of great things that are going to happen in the future. I want to be there. One of those things is first contact. Now, a lot of you might recognize this picture from the movie Star Trek, First Contact. And this is Zefram Cochrane, the future inventor of warp drive to go as fast as the speed of light. He's just gone faster, he's played by James Cromwell. He's just gone faster than the speed of light, and this alien species called Vulcans detected it, and they came to Earth to meet who it did. So he got off the ship, and he walks off the ship, and they shake hands, and it was a very powerful moment that I know it's not real in the movie, but this is going to happen someday, and when that happens, I want to be there. I want to take the time, I want to thank the coalition for the Radical Extension, and I want to say that I'm a member. I'm a member of the Coalition of Radical Life Extension. I want to say I'm also a member of the Transhumanist Party. Both these organizations are really important for getting us so that we can fight aging and death. And on behalf of Sierra Sciences, Defy Time, and Labella Gene Therapeutics, thank you.